everyone, and welcome to today's science video on the scientific method, where we will be looking at what the scientific method is, all its different parts, and how it can be applied. So let's jump right on in. The scientific method is a method of procedure that has characterized natural science since the 17th century. So it's consisting of systematic observation, measurement, and experiment, and the formulation, testing, and modification of a hypothesis. Today we're going to be looking at what all of those different parts mean and applying it to an example science project. So the first part of the scientific method is an observation. So this is the act of attentive watching, perceiving, or noticing. And so we make observations all the time without even realizing it. But when we're making a scientific observation, it's noticing something that might be out of sorts, or we're trying to explain why it happens. This will lead us to our next step, which is research. This is a systematic investigation into and study of materials and sources in order to establish facts and reach new conclusions. So we're looking for any information we can find on the weird observation that we just had. We're most likely not the first person to ever make this observation. So we want to see if there are other scientists or other people in the world who have made similar observations, have studied this before, and if they may have answers to the questions we already have. Then, based off of our observations and our research, we can make an informed guess as to what's going to happen. This is what we call our hypothesis. So the formal definition of a hypothesis is a scientific inquiry that can be tested, verified, or answered by further investigation or experimentation. And it's always in an if-then format. So this means if blank happens, then blank will happen. But we'll get into that a little bit later when we do our example. Based off of our hypothesis, we now need to test it. And we do this by making an experiment. An experiment is a procedure done in a controlled environment for the purpose of gathering observations, data, or facts, demonstrating known facts or theories, or for testing a new hypothesis or theory. This is going to have multiple steps, and you're going to need to follow these exactly in a row. Some of you may have already done some different science experiments, maybe at Paraclete or at your schools, but so this science experiment you are going to create to answer the questions that you have already asked in the earlier steps of the scientific method. The next part of the scientific method is analysis, and the point of analysis is to resolve anything complex into its elements for the purpose of the examination of each separately. So essentially you're going to be looking at what you found out from your experiment. What did the experiment tell you? And you're going to be analyzing it for the different information you can gather, and then trying to figure out if it can confirm or deny your hypothesis. The way you analyze your experiment will then lead you to results and conclusion. So the results is the interpreting of the test and deciding if the hypothesis should be rejected. So hypothesis is rejected if the results contradict it, showing that it is wrong. So if after your experiment, your hypothesis, the things that you thought were going to happen did not, then you're going to reject that hypothesis and most likely alter it or make a new hypothesis. And then we have our conclusion, which is a formal statement that can be evaluated independently by others, which is based off of your results. So these steps all make up our scientific method, but it's a lot easier to understand them when looking at them in the context of an actual science project. So that's what we're going to do next. I'm actually going to be going through what was my own 6th grade science fair project, and showing you how each part of the scientific method fits into the project. So to start, I made an observation. So my observation was that there's this thing called the 5-second rule, which I'm sure all of you are familiar with, which states that if food is on the ground for 5 seconds or left, then it's safe to eat. So then I did some research. So research, you always do research on topics related to the subject that you are studying. So I had to research about bacteria found in schools because I was doing my project. I was going to be looking at the 5-second rule in my own school cafeteria. So I was looking at bacteria found in schools. I was looking at other research done on the 5-second rule because there had been some studies trying to debunk it before. And I found some studies supporting it and some studies denying the existence of the 5-second rule. And so based off of my research and my observation, I then created a hypothesis. Although I can't remember exactly what it said, it was something along the lines of, if I drop food on the floor for 5 seconds, then it will pick up an unhealthy amount of bacteria. Remember, your hypothesis needs to be in an if-then format. And that's what that sentence was. If this happens, then this will happen. So next I need to test my hypothesis. And so that's when the experiment comes in. So this is going to be a very simplified version of how I conducted my experiment. But just so you get an idea, I started by dropping multiple samples of food onto the cafeteria floor for 5 seconds each. Then I swabbed the food and used a petri dish to collect the bacteria for each sample. For comparison, I swabbed a food sample that did not touch the ground at all, and that way I could see if the food itself would grow any bacteria, so that I could compare the food sample bacteria to the floor sample bacteria in my analysis. In this case, the food that does not touch the ground but is still swabbed is what we like to call the control group. In my case, this food is acting as a sample that will be compared against the samples that did touch the ground, so that way we have a baseline of which to regard the other one. So in an experiment, your control group doesn't go through the modifying factor, and so in my experiment, the control group of this sample did not go on the ground for 5 seconds. The next part of my experiment was to allow the bacteria to grow in the petri dishes for 7 days. Up next was analysis. So after the 7 days, I compared the control group sample to the other groups. I saw that the petri dishes from the samples that had touched the floor had significantly larger amounts of bacteria on them. So then for my results and conclusions, I could conclude that the 5-second rule is a myth. And if you drop food on the floor for 5 seconds, then it will indeed pick up an unhealthy amount of bacteria. I hope going through my 6th grade science project was helpful as far as solidifying the ideas of what is the scientific method and what are the different parts of it. I want to challenge you all to, over the next couple of days, see what observations you can make and brainstorm how you could use the scientific method to answer the question you have about that observation. It's a great exercise and a way to solidify the knowledge that you now have about the scientific method. Thank you all for watching, and keep working hard.